conference on our, this Monday rounds, which we always have. And so those online know that you ask questions using the Q&A feature, and those in the room, we're gonna pass a microphone or get you to stand up at the podium. But I am very, very pleased today to introduce Hee Cheol Cho from the Johns Hopkins Department of Pediatrics and Congenital Heart Center. He's here, not his first time in Ottawa, but first time here at the Heart Institute. And I learned last night that there's a very significant 6 degrees of separation here at the Institute. There are a number of our investigators and Hee Cheol who have actually trained together and who are colleagues and friends. And so in many ways, I think we're lucky not only to have an excellent speaker to come and share his research, but also in a way, sort of an extended friend of the Heart Institute. So thank you for coming. So I just wanted to introduce Dr. Cho. He's an associate professor at the Department of Surgery, division in pediatric cardiac surgery at Johns Hopkins School of Medicine since 2022. Prior to that, he was at Emory. And right now, he's the director of research at the Blalock-Taussig-Thomas Pediatric and Congenital Heart Center. And they lead pioneering research initiatives in pediatric and congenital heart surgery. Dr. Cho's research aims to understand the underlying principles of cardiac conduction, and you can see by his talk, today, we're going to be learning all about pacing and gene therapy. And his work integrates genetic engineering, stem cells, electrophysiology, and really it's understanding the mechanistic insights of the heart rhythm. The mechanistic project in this lab discover the forces that shape the exquisite architecture of the cardiac conduction system. And, the fundamental biology that he does in his lab is also linked very closely, of course, to translational research projects using preclinical animal models. We've talked about organoids and the future of organoids. And his goal is to make a paradigm that changes the impact on clinical outcomes for pediatric and congenital heart disease patients. He also leads a cross-disciplinary research team to extrapolate the bioengineering and design principles to tailor the therapeutics for cohorts of patients with cardiac arrhythmias. So as I say, we're not just fortunate to have Hee Cheol here to share his really pioneering and cutting edge research, but also as a friend of the Institute. So please, Dr. Cho, join us at the podium. Thank you for coming.
Thank you very much, Dr. Rayner. This is more than kind introduction. I'm incredibly honored and happy to be here. I feel like this is the first time giving a talk in Ottawa, but it doesn't feel like the first time looking at and meeting with our esteemed colleagues. And the first 3 hours that I spent on the second and third floor, I already saw incredibly energetic students and trainees, as well as the PIs. I think you have a lot of great things going, and so I'm so pleased. Without much ado, and I think it happened before, about 5 minutes ago, that it was slow advancing. Will probably happen. The next slide is my disclosure. Sadly, I have nothing to disclose, which means I don't have any company. Got it. Thank you so much. Yes, I need it. I'll tell you the motivation of the stuff that we have been doing, and mention a little bit about this somatic cell reprogramming approach that we have stumbled upon and expanded, and our most recent and earnest effort to make our discovery into a therapeutic modality. The cardiac conduction system, as we all know, starts from the sinoatrial node, which is really the conductor of an orchestra that dictates the rhythm and rate of our heart. And then it spreads around the atrial myocardium, arrives at the AV node, which is really the Rome. All the electricity arrives at the AV node. And there's a bit of a delay, about 120 to 140 milliseconds of delay, which is not by chance, but by design, that allows the blood to empty from the atria to the ventricles. After that delay, then it spreads down to the his bundle and Purkinje fibers, and the whole heart contracts through bundles and fibers. The anatomical discovery of the cardiac conduction system is actually backward. The Purkinje fibers were discovered exactly 100 years before my mother was born. And then his bundles were discovered, and Hawara, when he was in Germany, discovered the AV node, and the sinus node was discovered just a few years after that. The functional designation of the SA node as the pacemaker was actually a few years after the anatomical identification. When we look at the historical events, I think what we can learn from that is there are a lot more Purkinje fibers than his bundles, and there's only one AV node and one SA node. So maybe it was a little easier to discover the bundles and fibers. When things go really well, we don't need any doctor or treatment, but our heart rhythm and rate can become a little disorganized, and that's arrhythmias. It can be too fast, tachy, or too slow, bradyarrhythmias. And in both conditions, it gives us not enough time for the mechanical events of ventricular emptying and filling. If not addressed, the cardiac output drops, the lungs become congested, and the circulation collapses. It affects a lot of us, both Canadians and Americans. And there are a lot of people who eventually succumb to the cardiac arrhythmias. And it's expensive. And in the US alone, and that was, I'm sorry, I haven't updated, more than 10 years ago, that every year about $3 billion are paid to healthcare insurance programs in the US alone. The treatment to cardiac arrhythmias are several layers, and the drug therapy works sometimes, and a lot of drugs actually invite new arrhythmias to begin. And we can also get ablations, radiofrequency ablations to burn some specific sites. It can be potentially curative, but a little difficult for more complex arrhythmias such as atrial fibrillation. When all things fail, and in the case of too slow heart rate, bradyarrhythmias, the only thing that we can get is an implantation of a pacemaker device. And here's a typical pacemaker device on the top right corner, which is really about 80% battery, 20% electrical circuit. And pacemakers, at its essence, it's an electrical generator. It sends electrical shock. And you need that wire to go through your blood vessel, and that wire gets implanted into the heart. There are about 300,000 pacemakers implanted every year in the US alone. Globally, it drives and fuels about $5 billion pacemaker industry. When I need a pacemaker almost all the time, I will need a pacemaker for life. There are complications with leads, generators, or both, mostly the problems with the foreign hardware in the body. There are leadless pacemakers that doesn't have any of these electrical wires that go through the blood vessel, but leadless pacemakers, the whole thing needs to go inside the heart. And most of the time, these leadless pacemakers are implanted for patients who are supposed to have less duration for pacing, for elderly. The infections related to the pacemaker devices is on the rise. It has been on the rise for some time, but now that more patients are getting the pacemakers, the actual case numbers are high. So the AHA has a mandate to find ways to reduce infections. When the pacemaker is infected, most of the time you have to take the whole device out for the antibiotic treatment. And you need to have a secondary device to pace during that infection control phase. All these problems get amplified in pediatric condition, and this is a surgery in I believe it was a 2-year-old boy, where babies are small. There's not enough space for the pacemaker generator to be implanted in the pocket. The blood vessels, or the vasculature, is too thin for the lead wires to go through. So what happens is you open the chest. For most pediatric applications, the generator is implanted in an abdominal cavity, and the wires are run through the chest, and the pacing wires are attached or sutured on the heart surface. Babies grow wires don't stretch. So in a lot of times, the surgeons or cardiologists want to leave more wires into the body, but the wires get tissue adhesions. So even when there's extra wires that are implanted, it is not unusual for these patients to undergo 2 to 3 fairly extensive and major cardiac surgeries in the span of their first decade in life. So the current strategies for cardiac pacing in pediatric patients, particularly, is far from ideal and often inadequate. And that's one of the biggest motivations. And what are the advantages of biological therapy? Well, a biological therapy with cells and genes and cells are the nature's nano and micro particles. So they're so small, and we should be able to deliver them in a more deterministic manner. So we could highly localize these genes in a way that we want to. When we use especially catheter-mediated delivery of these genes or cells, the surgery can be minimal. It could be a femoral vein access to go to the inside of the heart and deliver the gene. And in some cases for gene therapy, once the therapy is delivered, there's almost no footprint for the therapy that resides in the body. And these are really the conceptual benefits where a biological therapy should be able to rate on that. When I want to catch the bus and run to the bus stop, the heart rate should increase. When I sit down to relax, I don't want my heart rate to be 120 beats per minute. So the biological therapy should be able to rate on that, manner that our heart rate adapts in natural physiology. As a safety measure, we also want the biological therapy to be conventionally reversible by conventional approaches such as ablation to burn it if we don't need that. Durable, and at the end, it needs to improve the clinical outcomes. In 2002, when I was toiling away and patching on cardiomyocytes in downtown Toronto doing PhD, there was a paper published by a group in Baltimore on a topic of biological pacemaker published in Nature with a single figure. And that really made me both excited and unhappy because they were working on a channel that was my PhD project, which is called KIR 2.1. And it is an inroad rectified potassium channel. And the idea was to make a mutation of the channel and downregulate an ionic current called IK1, destabilize the action potential of a myocyte, and thereby allow ventricular myocytes to pace on its own. And that was the idea. And I'll just go through the biological pacemaker concept back then, which is change or alter the action potential profile of a ventricular myocyte to something that looks like the SA node, which is very easy to see on a PowerPoint slide. And this is what I meant by reducing IK1 using AAA mutation of an KIR 2.1 inward rectifier potassium channel, thereby destabilize this resting potential and allow the cells to pace on their own. A lot of work was done. That was my postdoc project. Group in Colombia also attacked this approach by overexpressing an ionic current called funny current, which is called IF. And really, there are two different things that differentiate a ventricular myocyte action potential and the pacemaker action potential, in that the pacemaker cells do not really rest, as in the ventricular myocyte. And the pacemaker cells have slowly and spontaneously depolarizing membrane potential, aided by this funny current, as well as calcium currents. Fast forward a few years, and when I saw that paper in 2002, I wanted to get into this research field, apply for postdoc position in Baltimore, and studied my work. After a few years, and I think I will get to talk with some of the trainees after the talk about what the life I led, mostly failure. Um, and I actually, after my short postdoc, I worked for a startup company. I drove the company to the ground. And during that time, I learned, well, there's this new technique called IPS. You can convert a very boring cell like skin fibroblast into a cell that looks like a stem cell. And that was during my company time, reading papers and fantasizing, maybe we can do something. And this is the sinus node um, from a red heart, and it is really an autonomous organ within the heart. You can think about the sinus node, the pacemaker tissue of the heart, as the organoid within the SI node, because you can dissect it out. You can keep it in a cell culture, and without any technique, you can keep it beating like that for days. And we wanted to benchmark the SI node rather than just changing an action potential by suppressing IK1 or increasing IF. We wanted to see whether we can actually create the SI node cell. Sinus node is really small. I mean, it's astounding. It's sort of awe-inspiring that there are less than 100,000 cells in our heart that drives in each heartbeat just before we are born, way before we were born, and until we call it a day. And the structure is very interesting. These cells are not organized. When you look at the cell orientation, it's isotropic. There's no sense of direction. There's a lot of fibroblasts in the sinus node that if I don't tell you that this is sinus node, you will look at the histology and say, this is a heart failure because it's so fibrotic. And that's the natural SI node. The cells look a little different, and I'll show you some images of that. And I'm not going to get into this part, but in the past 15 years, the mechanism or the ionic biophysical mechanism of the pacemaker cells has been fairly well studied. And there are two clock mechanisms, one mediated by ion channels in the cell membrane, and the other one is the SR calcium release events that happen spontaneously and autonomously, unrelated to the ion channels there. So there are membrane clock and calcium clock mechanisms. Here's one of the more famous figures from Waddington back in 1957. And here is a stem cell. We all start from there. And then over the course of major steps of development, the stem cell will become a terminally differentiated somatic cell. The IPS technology will take that terminally differentiated cell and back into the IPS. And then some of us in the field derive cardiomyocytes of the atrial ventricles and sometimes into the pacemaker lineage back down the developmental path. And Dr. Protzo, sitting here, is really the champion of these human IPS-derived pacemaker biology and application of that into cardiac pacing. What we thought was maybe we can go from one type of cardiomyocytes to another type of cardiomyocyte in a direct manner without going to IPS. And the motivation was maybe it would be easier to go from a myocyte to myocyte, a ventricular myocyte to pacemaker myocyte, rather than going from a fibroblast to IPS. We studied a lot of developmental papers back in the late 2000s and early 2010s to understand that there are a handful of transcription factors that are important for the development of the SA node. And these are studies performed by Vincent Christopoulos in Amsterdam. And TBOX18 is one of the factors that reside in the sinus horn and later becomes the sinus node. In fact, if you delete both alleles of TBOX18, you lose about 70 to 80% of the SA node. And other studies that showed importance of some distinct transcription factors in the development of the SA node, we looked at them and we thought we thought, well, we'll take each of these genes and test them. And our idea was, well, Yamanaka had to have 4 factors to create IPS. We'll probably have to combine a couple, 3 or 4. And the idea was to take TBOS18, TBOS3, SHOX2, TBOS5, and a few other factors and test them one by one. Our most used in vitro system is cardiomyocytes that we isolate from a newborn rat heart, because these cardiomyocytes are culturable, unlike adult cardiomyocytes. And we added these genes one by one. We were surprised at the beginning because we thought we would have to combine some of them, but right off the bat, we saw that TBOS18, when it was added into the neonatal right ventricular myocytes, NRVMs, made these NRVMs beat spontaneously. Now, there is a disclaimer. These neonatal right ventricular myocytes are immature. They're newborn cardiomyocytes. They're cultured. So they do have some spontaneous activity. So as you can see from the N numbers, we had to sort of, at the beginning, test a lot of conditions to make the control condition a little quiet. And a little quiet would be about 30% of these monolayers that we were testing were beating spontaneously. But when we added TBOS18, a lot of these monolayers began to beat spontaneously. And here's another sort of visual manifestation that when we added a jellyfish gene, green fluorescent protein, as a control into the neonatal ventricular myocytes, we saw some cells that would have spontaneous activity. And this is a calcium cycling, and this is not being paced. So what you see are the cells that cycle calcium on their own. When we added TBOS18, we saw fireworks. And this phenomenon was easy to see. And we can create this phenomenon on a weekly basis now, but at the beginning, we had to learn that the dosing was actually very important, that when we added a lot of viruses, it actually ended up killing these cardiomyocytes. We had to dial down the dose of TBOS18. And we also looked at the conduction velocity, which is really how fast electricity spreads from one end to the other end. And I mentioned in an earlier slide that SA node has a slow conduction velocity compared to the ventricular or atrial myocardium. For people like us who do cardiac electrophysiology on the bench, it's an important phenomenon because the SA node is sitting right in the smack of middle of atrial myocardium. And it's surrounded by atrial myocardium that has minus 85 millivolts of very low negative resting membrane potential. If it doesn't have any mechanism to protect itself, it will not be able to pace the atrial myocardium, but instead will be suppressed. So the one way to achieve that, and it's called a sourcing mismatch, where electrical source of the SA node is no match for the large electrical sink of the atrial myocardium, is to have very weak cell-cell electrical coupling within the SA node. And that is recapitulated when we delivered TBOS18. It reduced cell-cell electrical coupling and slowed conduction velocity. And that was due to basically shutdown of connexin 43 from control to TBOS18 transduced cells. And another thing that's peculiar about the SA node is that CX43 expression is almost devoid in the SA node. Instead, it expresses a very low gap junction connexin called CX45. Here is the in vivo experiment that we did. We delivered TBOS18. It had a reporter gene, GFP. So we would inject a rat or guinea pig myocardium by direct injection with TBOS18. A few days later, we will take the animals back. We use ex vivo perfusion system to digest and isolate live cardiomyocytes and look for green cells, which is a reporter for TBOS18. And here is the first data that we saw that really surprised us, because it was a phenomenon that we didn't look for but came to us. Some of the cardiomyocytes. So left, this is cardiomyocytes that are differentiated from animals injected with GLP alone. And on the right, on the middle, this is cardiomyocytes isolated from animals injected with T-VAS. The ventricular myocytes of the guinea pig or the rat heart is sort of rectangular shaped, rod shaped with very well organized sarcomeres. The T-VAS18 expressing cells, again, T-VAS18 is a transcription factor, so its expression is retained in the nuclei. Some of these cells actually lose some weight and become have tapering ends, which is really similar to the gold standard. These are 1, 2, 3 cells that are isolated from the synovial node of the same animal. So it's a phenotype that we actually see quite often, but we still do not understand. And we're actively trying to investigate what kind of remodeling that happens in the ventricular myocytes that makes the morphology of these ventricular myocytes towards more nodal phenotype. But what made us the most excited was the PSI. Ventricular myocytes expressing a control gene is basically a no-show. It sits at around -80 millivolt. It fires an action potential only when we inject a little bit of positive current and stimulate it. But once we trigger the action potential, it stays quiet. And this is an expanded time scale. Some of the ventricular myocytes, not all of them, that express T-VAS18 showed oscillating action potentials that almost look like the oscillating action potentials from the gold standard, the synovial node. And this data is acquired by no other than Dr. Wenbin Liang when we were the superstar team at Cedars-Sinai working together. And I have to say that we have collected and tested more animals in the past 15 years. And I still show your trace because this is one of the most beautiful traces that we have seen. We also looked at the cell level, whether the cells will be able to beat faster or slower with beta-adrenergic stimulation or cholinergic suppression using isoproterenol or acetylcholine as the reagents, and their beating rates all responded. And here's the in vivo data back then when Dr. Liang and I were working together, and we used the best animal data model that we had back then, which was not the best model, and I'll be able to show you some other models in later slides. The idea was to block the AV conduction using a chemical method calling. But before we do that, we actually inject our gene, either GLP gene at the apex of the guinea pig heart or T-VAS18 in another set of animals. And then chemically, we slow down the heart rate, and eventually the animals go through AV dissociation, which means the sinus rhythm is relatively okay, but the conduction through the AV node is now disabled. And in control animals, you see the ventricular beats in the form of QRS complexes. And then you see the atrial beat in the form of P waves that are not coupled. And you have basically 2 sets of drummers, a drummer from the SI node and a drummer for the slow, we call it escape rhythm, that's mediated by slow pacemaker cells in the his bundle area, AV junctional area. In animals that were injected with T-VAS18, what we saw was faster QRS complexes in these animals. Also, the ventricular, the polarity of the QRS was opposite. And injection into the apex is by design. We wanted to see that if the conduction happens from somewhere here, as in control animals, on lead 2 of the cage, it will be antigrade or the normal conduction, and the QRS will be positive. If pacing comes from the opposite side, then we'll see a change in the polarity of the QRS complexes. So that's what we saw. And we also reasoned in these animals, small animals, there's not a lot of engineering that we could have done to create conduction using cardiac conduction system. The spread of electricity from his bundle Purkinje fibers is really fast and QRS is very, very narrow. When the conduction slows down, you will see widening of the QRS complex. So these were the two lines of evidence that we saw to understand or have some faith that this pacing was actually coming out from our gene injection. And from that most important sort of founding study back in 2013 by Nidhi Kapoor and Dr. Wenbin Liang, we have studied other mechanisms and cell biology of this TBOS18-induced pacemaker reprogramming. In the past 5 years, 7 to 5 years, we have now engaged into understanding how not only create pacemaker cells, but create a pacemaker node. And one of the things that we understand is pacing comes from not cells but from a signature node. And another thing that we, during earlier studies, we saw, but we didn't really get into it. But the more we did the studies, we got to appreciate and acknowledge is that when we delivered TBOS18, the amount of fibrosis at the injection site was quite severe. So this is control animal at day 4 or 2 weeks after injecting GFP. And you can imagine these are rat hearts and guinea pig hearts beating at around 300 beats per minute. You advance the needle and inject your gene into the myocardium, we're going to create injury. So there's a mechanical injury there. We're injecting adenovirus of a transgene, so there's inflammation. So that's what we see in control animals. But what we saw with TBOS18 was above and beyond what we saw in control animals. And then we became a little more serious about to understand what is this fibrosis about. And there were both pro and anti-inflammatory signaling that was happening when we delivered TBOS18. And a lot of them were interleukins, as well as TGF-beta related signaling. And fast forward a little more, we performed proteomic studies and transcriptome studies to really pinpoint that at least there is a TGF-beta involved inflammatory or fibrosis activation that TBOS18 triggers. And what we feel today is that this TGF-beta activation and fibrosis may be a real thing. Maybe it's something that the SA node really needs through development. Remembering that TBOS18 is not a disease factor, it's a transcription factor that's really important for the development of the SA node. And the SA node is really fibrotic compared to the natural myocardium. In the developmental context, this may be one of the natural things that TBOS18 does. But in our context of postnatal heart or cardiac pacing with gene therapy, TBOS18 may be doing this in an overblown manner. So we wanted to soften that TGF-beta signaling. We tested a few different drugs. And here is a drug called A8301. In control animals, with and without the small molecule, it didn't suppress a lot because this was sort of a baseline fibrosis that we saw due to mechanical injury that we caused by needle advancement. And in TBOS18 animals, we saw reasonable degree of softening of the fibrosis here. And this is all has not been published yet. And we also wanted to look at this in vitro to see whether there is any pacing differences in our cardiomyocyte model. And a little after, and this takes a bit of eye training, a little after 2 weeks, these cardiomyocytes do beat. I'm not sure if you can appreciate that the cells are contracting, but they're really not beating together. And that's what we see over time when we deliver TBOS18. At an earlier time point, all these neonatal right ventricular myocytes beat together. but that synchronous pacing becomes undone by the end of week 1. And by week 2, this is what we see. And this is the same neonatal left ventricular myocytes with TBOS18, but with TGF beta inhibitor. Not all of them, but some of these cardiomyocytes now begin to be together, and their pacing is synchronous over time. And when we looked at this phenomenon on multi-electrode array to get more of a quantitative data, we understood that TBOS18 induced pacemaker cells beat at week 1. By week 2, the number of beats that we see on multi-electrode array decreases, and probably because, maybe not because we're losing the pacemaker cells, but because they lose the ability to beat together, and multi-electrode array cannot register that asynchronous pacing. When we add a TGF beta inhibitor, we can maintain that synchronous pacing. And we looked at this phenomenon in vivo in a rat model of AV block. I mentioned that back about 10 years ago, we didn't really have a good model of AV block in ambulatory animal. Now we created one by using an acupuncture needle to advance that acupuncture needle into the AV groove, and then pass through a electrical current through that needle to burn the AV node area. Now, when we do that, the animals, their heart rate drops on the spot from around 300 beats per minute to about 140 beats per minute. Some animals survive, some animals don't. The attrition rate is pretty big. Unfortunately, most animals that survive are female rats, not the male rats. So there's some unfairness in that animal model. In the past 18 months, we actually refined that model. So this is a head-to-head comparison. We're actually using a radio frequency ablation catheter to burn the AV node from the outside on the epicardial surface. This was our previous model where the attrition rate was more than 50%. When we start with about 24 to 26 rats, with our newer model, our attrition rate is much better, with about 80% of the animals surviving. This is important because when we do small animals studies, if we set out to do 10 animals, get 10 N of 10 data, with this model, we have to start with about 30 rats to get enough power. And this is also under review in a journal called Lab Animals, which is sort of based on animal model creation. It was a 1-month study to create AV block, and then we gave about 1 week to understand and save these animals that survive the complete AV block and severe bradycardia. And then we inject our gene of interest, and also have an osmotic pump that releases the small molecule that inhibits TGF beta. And here is a preliminary data where we saw that TBOS18 animals co-administered with a TGF beta signaling have a sustained ventricular heart rate that's above and beyond the control. Animals were injected with GMP with a compound, or TBOS18 animals with a placebo. So we're really excited about this adjunct that really sustains the TBOS18 pacing. And here's the last chapter of this presentation where we have taken what we know today and developed this as a therapeutic modality. And back in 2018, we were thinking about, okay, so we know something about TGF beta signaling. It seems like we have found one way to make this pacing last a little bit. And are we going to inject adenovirus? And my home department is in pediatrics, and gene therapy has a sort of bad memory many years ago about gene therapy in the first phase. So I wasn't really fully comfortable with adenovirus gene therapy. So we looked at synthetic mRNA. because about, I think, 2 years before 2018, 3 years before that, we saw a paper by Ken Chen and their Grassi from Boston, but they used mRNA for VEGF for cardiac regeneration and vasculature. So we were emboldened. We looked at, okay, we will look at the mRNA as a potential gene therapy because we may be able to sidestep all the problems with adenovirus-related inflammation. Maybe less immune and inflammatory response compared to viral vectors for the mRNA. mRNA doesn't modify genome unless you use CRISPR mRNA. And mRNA degrades pretty fast, so it doesn't leave any footprint. And here's one data that we looked at that gave us more motivation. When we looked for TBOS18 in the developing sinus node at mouse embryo day 16.5, the TBOS18 positive nuclei in the sea of HCM4 positive cells were there. We could see them. About 3 days after the mice were born, we didn't really see much TBOS18. And in the developmental biology field, it seems that TBOS18, just as we have demonstrated in this immunostaining, TBOS18 in the mouse life appears to be a transient factor that after birth, it is no longer expressed. I just learned this morning from Dr. Kim that in human heart, TBOS18 is actually expressed throughout life in the postnatal heart test model, which is very, very interesting. When we looked at the TBOS18 as an adenovirus vector in NRBMs and in this Western blot of time course, we also saw, for reasons that we cannot explain today, TBOS18 protein expression with adenovirus also goes away in about a week or less than 2 weeks. This is here. I think I will just say whatever we think today. There must be some negative feedback loop that's inherent to TBOS18 that turns itself off, and we have some aspects that we are studying in that domain. Seeing that TBOS18 is not expressed, it's a transient vector in the mouse, let's say. Seeing that with adenovirus, the longest that we express is about a week or 10 days, we thought, okay, if the mRNA can compete with adenovirus gene expression pound for pound, here's adenovirus GMP on cardiomyocytes, in vitro transcribed GMP on cardiomyocytes, and our 7 messenger RNA derived GMP kicks in really fast. By hour 6, we can see that adenovirus catches on. And when we look at a 16-hour time frame, by hour 7, messenger RNA mediated gene transfer has already reached the peak. Whereas with adenovirus, it's still on a lag phase. Over the course of 2 weeks, though, mRNA-based gene transfer goes down within about a week, whereas adenovirus gene expression peaks a little after 1 week. So we thought, okay, well, 1 week expression with a synthetic mRNA technology back in 2018 may be good enough. We took that mRNA, and another thing, back in 2018, all the literature with lipid nanoparticle was, ooh, cytotoxic, ooh, maybe inflammatory. So we weren't sure whether we wanted to complex our mRNA with LNP, although LNP is known to really enhance the transfer of your nucleic acid. So we opted not to use LNP, but just inject mRNA straight into the heart using saline. And that's what we did. And we mixed some near infrared dye for eye candy. So here's mRNA that we injected into the heart. It was a rat heart. And the idea was, oh, we've got the mRNA in saline. Does it actually go into the cardiomyocytes? We brought the animal back the next day, harvested the heart, sectioned it, looked for GFP, and lo and behold, like some of the mRNA actually goes in. We didn't do it from looking for a jackpot. We were working with Phil Santangelo at Georgia Tech when I was at Emory, And Phil Santangelo was already working on delivery of mRNA in saline into the lung tissue, liver tissue. So that's what we have tested, and we were emboldened to use mRNA in saline. And here's biodistribution study, where we looked at mRNA luciferase directly injected into myocardium, and we wanted to see how long it lasts. And it lasts about 5 days. And this is average data of luciferase expression in vivo, in the heart. Biodistribution. We also wanted to understand, although we deliver the gene directly into the myocardium, would any of these genes go astray and go to another organs? And here's biodistribution of adenovirus, luciferase, and biodistribution of in vitro transcribed mRNA. At the heart, we see these. Each dot is one animal. And in the liver, lungs, spleen, kidney, and intestine, the biodistribution of the mRNA or off-target expression of mRNA was more favorable than that of the adenovirus. Probably because in systemic circulation, mRNA degrades so fast. Here's the pivotal study. We called it BioPace. It's TBOS18 mRNA with TGF-beta inhibitor in a porcine model of complete heart block. And this is the system that we have established. And day one is pretty long. The first thing that we do is actually we implant a pacemaker. And here's a pacemaker lead. We call it is a right ventricle pacemaker where we advance the lead pacemaker lead wire into the RV and secure it onto the RV apex area. Once we implant the pacemaker, we then go in with a catheter, radio frequency ablation catheter, and burn the AV node. And the reason for the pacemaker lead or pacemaker is that as soon as we burn the AV node, the peak heart rate drops from around 80 beats per minute to sometimes less than 50 beats per minute, and we may lose the peak on the table. So we wanted to keep the peaks alive, and the pacemaker can be programmed as a backup pacing so that if the heart rate drops below 50 or 55, the implanted pacemaker will kick in and maintain the heart rate at the minimum heart rate of 55. That's 1 and 2. And then we change the catheter and go inside the heart and inject our gene, TBOS18 mRNA, into the high His bundle area. In the cartoon, we had needle going right to the His bundle. We don't have that accuracy. We injected it into the high His bundle area. We can sort of feel it because the His bundle, again, the conduction velocity there is really fast. And on the electrogram, it shows as a pencil sharp QRS. So we can reasonably sort of go into that area and inject. And here is the injection catheter. And maybe with a sprinkle of imagination, you may be able to see a little bit of shadow that comes out. Right now. Yeah. So that's not because mRNA you can see, but we actually complex our solution with a contrast agent so that we can visually confirm that injection was okay. Because you can imagine it's a beating heart, and we put the catheter in. Although we advance the needle, what we inject may come out right away. And we actually do see that some of these injections, we see contrast agent for 1 heartbeat and it's washed out. Then we have to inject it. So that's one way to reduce non-responder. Here is a 4-week study that we have done. And here's a data where controlled animals injected with GFP. This is a 3-day ECG heart rate data. The backup pacemaker rate was at 55 beats per minute. And most of the time, the control pig was being paced by the backup pacemaker. There was a little bit of faster heart rate sometime in the morning. Most of the time, it was higher. TBOS18 injected pig instead showed an acceleration of heart rate sometime in the morning. And this is 24 hours. It was sustained for morning and early afternoon and slowed down. The next morning, it did the same thing. And the next morning, it did the same. When we first saw the data and we had a real-time data feed in from the animal facility to our lab, it was around 9 or 10 p.m. We were like jumping up and down because, wow, this is different. And then another thing is what's happening at 8 a.m., 9 a.m. We called the animal facility the next morning to understand what's going on. The animals are fed, the pigs are fed at around 8 or 9 a.m. And that's the only time that they are fed. So you can imagine the pigs are excited. The pig cage is no bigger than about this area. So they don't have a lot of room to run. But the anticipation of the food and seeing the food and eating the food will get them so excited. This is not the exercise. This is emotion. Imagine biological pacemaker responding to the emotion of the pigs and the patient and the heart rate increase. It is something that we didn't really appreciate before we did the experiment, and we got to understand. And if any one of you walked into a pig room, you will know when they eat, you cannot walk in like this. You have to close your ears because it's loud. And over the course of 24 days, what we saw was the same sort of heart rate acceleration in the morning and then slow down when they go to bed or sleep in the night, the heart rate decreases. So we're incredibly excited about this data. And here's we also had a telemetry that records ECG 24/7, but the telemetry also has an accelerometer, which is in our smartphone. So it can actually track activity. The pigs, when they move, they don't do that a lot, but when they move, you can track them. And the solid line is heart rate over the course of 24 hours. And the activity is the dotted line. Control animals show this. T-box18 injected animals show heart rate change over the course of 24 hours. And the activity sort of goes hand in hand with the heart rate change. So what the data tells us is that the biological pacing can rate adapt with the animal's movement. And here's an anatomical mapping that we performed to sort of understand if the pacing is actually coming from somewhere close to where we have injected, which was sort of the side of the heart, high septal area. Here's a conclusion. We have tested a synthetic mRNA as a gene delivery vehicle for T-box18. It creates cardiac conduction system pacing in a porcine model of complete heart block for at least 1 month and ready for temporary pacing needs. We say ready for temporary pacing needs because not because it's not useful after 1 month, but because we have the 1 month studies, what we have done so far. We are actively pursuing or performing the 3 months efficacy study today. And David Wolfson was really the lead on this study. So here I mentioned about the potential advantages. We have shown that localization is possible with gene therapy. It was a minimally invasive right femoral vein catheterization, no open chest, leaves no footprint for the synthetic mRNA part, and the biological pacing can rate adapt. And not shown in this slide deck, but we have gone in and used a radiofrequency ablation catheter to burn the injection area and take out the biological pacing to show that it is reversible. We're looking into an IND-enabling preclinical studies for 3 months efficacy, and we're actively working with cardiologists and regulatory bodies to design our IND-enabling investigational new drug enabling, which is a prerequisite to get FDA approval. And it's coming to a cath lab near you. And that's what I usually say for our clinical colleagues. The most important slide is really here, and really talented and devoted individuals in our team and collaborators. I'm basically a cheerleader for all these friends and colleagues on this platform, and really grateful for all the funding support for this study. Thanks so much for your attention.
That was fabulous. Thank you so much. I've got a couple of questions myself. I will open it up if any questions from the audience, and maybe I'll start. The mRNA data was fascinating. Really surprising how much expression you can get by simply injecting RNA. And I mean, I think you're absolutely right about the biodistribution and why you wouldn't see RNA maybe, you know, elsewhere in the system. But I mean, just naive question. I mean, you know, how widespread is this? Is something about the heart in particular? There's lots of immune cells in the heart, of course. So even seeing this RNA, I would think it would cause a little bit of an activation of the immune response. Maybe not a bad thing, you know, maybe from a sort of remodeling point of view, it might be okay to tickle the immune system a little bit in this scenario. So just kind of your thoughts on that.
Fantastic question, and very important question. You know, this is a gene therapy which is a little different from the vast majority of the gene therapy in the heart, where the vast majority you want to express the gene, you know, in a large area. For cardiac pacing, you don't want that. You want the pacing come from a focal and a predetermined location. What we see for the spread is about 2 to 3 centimeters from the injection site, which is not as small as we. And we have 2 studies, 2 experiments, really, at this baby step level to find a way to confine the gene at the injection site, biomaterials. And before I go to the immune inflammation, I also want to mention that although we use saline for this mRNA gene therapy, COVID changed everything. So I mentioned back in 2018, LNP was a scary thing for us. But having received the vaccines that has mRNA with LNP complex, then we just got a sore arm. It changed our thinking. So we are actively pursuing complexing the mRNA with LNP. I think the road ahead for us is not to develop a brand new LNP. We are going to use either the Pfizer or Moderna, the SM-102 or ALC-135 formulations. It's all known, published. So we will use that. We have tested SM-102 LNP with our mRNA gene delivery in the heart, and the fold increase was 157-fold increase of gene transfer. It's astounding. So I think with the enhanced gene transfer with LNP, we should be able to reduce our dose, which is a big deal for a potential the envisioned clinical trial. The immune profile, we looked at it in our Wolfson et al. Nature BME paper, that we looked at a few panels, and you know, we picked things that will be good for us. All right. So for when it was really not a fair comparison between adenovirus and mRNA, and a lot of CD related vectors were expressed lower with messenger RNA gene therapy. But the mRNA also triggers the innate immune response as well. So I think it is important to understand that. And Daryl asked me yesterday at the dinner about the study from our friends and colleagues at Cedars-Sinai who discovered that TBAS18 mRNA injection or expression in cardiomyocytes elicits microRNA expression, and it actually turns off TBAS18. And this is, I think my understanding is really related to the chemistry of the messenger RNA, that because they saw a similar thing on using not TBAS18, but another transgene, but using the same chemistry. So I think some of the innate immune response and inflammatory signaling need to be studied based on the chemistry of the mRNA, which is actually not a simple thing. I realized we have at Hopkins, a RNA Innovation Center that just started about 8 months ago. And we're looking at different chemistry for messenger RNA. And it seems like the immune and immune inflammatory response can be fairly different depending on which chemistry you use. Very important ones.
Okay. Oh, okay, sorry. Dr. Cho, thank you so much for another amazing talk, as always. So I just have one question. Regarding the mechanisms of the fibrosis in the TBX18 expressed myocardium, do you think it is the direct effect of TBX18 expression in the fibroblasts, or it could be the paracrine effects of the TBX18 expressing cardiomyocytes?
That's a million-dollar question. I think we understand a little bit. We've done, so for reasons that I don't know well studied, both adenovirus and mRNA, I showed the data, but I didn't mention it. They go into cardiomyocytes much easier than they do into fibroblasts. So when we do the gene transfer with adenovirus or gene transfer with mRNA, most cells that are transduced or transfected are cardiomyocytes. What we have done was a simple, two-story culture system of Boyden chamber system where we had upstairs cardiomyocytes that express TBX18, downstairs cardiac fibroblasts. And it was clear that the cardiomyocytes that express TBX18 mobilizes TGF-beta, which then activates the cardiac fibroblast, and they become fibrotic. So that much we understand. If TBX18 goes directly into the cardiac fibroblast, of course, I think it will do the same thing. But I think just from the inherent gene transfer efficiency, it is really myocytes that express TBX18 that triggers TGF-beta. Then fibrosis comes from the cardiac.
Okay, thank you so much.
But there's just adding 2 more lines there. The cardiac fibroblasts, again, this is data-driven thing because I didn't want to study fibroblasts too much. However, it was important. So we have another unpublished but ideas. We wonder whether activation of fibroblasts is actually a bad thing. We have a hypothesis that it could actually help pacing because cardiac fibroblasts and myofibroblasts, the quiescent ones and activated ones, have different electrophysiology. And their membrane potential is actually quite different. More depolarized for the myofibroblasts. When you have, and this is shown by Stefan Rohr in the context of arrhythmia, that when you have myofibroblasts surrounding cardiomyocytes, it will depolarize the myocytes membrane potential enough that they will begin to trigger activity. And in the context of pacing, maybe it could be a help. So we have a project on that inflammation in fibroblast bias. Thank you.
Amazing talk, Dr. Cho, and I'm really happy seeing going into the PIC model and going to an IND enabling studies. It's all really exciting for the field. I think what I always wonder about, I love the whole reprogramming idea. How stably reprogrammed are they really? And now you have data showing that the TGF-beta prolongs it in the PIC, you seem to have pacing for months. But even in vitro, when you keep these cells, like your rat neonatal cells, have you looked after 2 weeks, a month, 3 months, how they compare to an SA node cell? Have you maybe done single cell RNA-seq and really compared them? How sure are we that really reprogramming and they stay stable even if TBX18 goes away?
Absolutely important question. We looked at single cell RNA, single cell transcriptome in vitro, days 3, 7, and 14. The reprogrammed cells emerge by day 7. The percentage of reprogrammed cells hold steady at day 14. However, the expression of TBX18 transgene that we deliver into the cardiomyocytes go way down by day 14. So we understand that presence of the transgene is not required to maintain TBOX18 reprogrammed cells at day 14, but that's day 14 and that's in vitro. I think what we need to do is in vivo. We have also looked at in vivo up to 6 weeks after gene transfer, but this is qPCR, and this is a methylation pattern that we looked at. And it seems like by at least at 6-week mark, the methylation patterns look similar in terms of more open structure for the HM4 and closed structure for the chamber cardiomyocyte contractile. So that's all the data that we have today. Now, so we'll have to do longer term single cell biology to understand how permanent the cell fate changes.
Okay. So thank you very much, Dr. Cho. And as a person who's teasing you when you're doing patch clamp in a long time ago, so it's great to see how IK1, story all the way to TBX18, to pacemaker story. So my question is actually, I was excited to see that recently programmed pacemaker cell is actually respond to autonomic nervous system. So what's your insight how they actually integrated to respond to autonomic system because of likely, maybe there's innovative nerves within the heart? And second question related to that is, do you think that reprogramming can be strengthened by the innovation and also interaction with the neuronal signals?
Oh, it's a question that we want to understand. There's no innovation. We looked for it at 2, 4, 6-week mark at the injection site. We did not see any innovation. I think the neurohumoral response to the physiology comes from the cell surface receptors that these cardiomyocytes, the nodal cardiomyocytes, express the beta-adrenergic receptors and cholinergic receptors. So, however, does it mean that there will be no innovation? I'm not sure. We're studying some of the chemoattractants, such as semaphorins, SEMA3A, 3C, these things to understand whether there is some inherent ability for the nodal cells to attract innervation. And it's one part that we really cannot advance on our own. We need to hold hands and run together with neuro people to co-culture and understand the innovation part, but this is absolutely important.
Thank you.
So much. I'll just double check online. There are no questions. I don't think, yeah, they're good. Okay. So thank you so much, Dr. Cho, for coming and visiting. And I know right after this, I believe you are having a mentoring session with the trainees. So for anybody who isn't aware, there's a trainee session with Dr. Cho in the multimedia room at 1:30. And I can just say, even just from chatting last night and today, I think it's going to be really informative. A Canadian trained export of the academic system in Canada. I think you'll have a lot of really interesting insights for our trainees. So thank you once again for the excellent presentation, and thank you for accepting our invitation to visit the Heart Institute.
Thank you so much. It was my great honor. Thank you.